much greater concentration gradients for the movement of oxygen from the lungs to the blood, because it has to compensate for the lower solubility. So we have to have greater pressure gradients to support exchange of oxygen because of its lower solubility relative to carbon dioxide, and we can get away with smaller pressure gradients to facilitate exchange of carbon dioxide from the blood to the lungs because it is a much more soluble gas, relatively speaking, than oxygen. Okay? So, if we look at the transport of oxygen and carbon dioxide along their pressure gradients, so here we're looking at the inspired PO2, as I showed you, of about 160 millimeters of mercury. So oxygen is inspired through these conducting airways into the trachea, and you can see that we automatically kind of desaturate that partial pressure of oxygen. As I mentioned, this is because this air is becoming warm to body temperature and humidified. And so as a result, we're getting a decrease in the partial pressure of oxygen within that gas mixture. So then oxygen then will transport it. You can see now it has a PO2 here of about 150 millimeters of mercury, but in the alveolus it's about 100. And so the alveolar PO2 decreases to about 100 millimeters of mercury due to continual oxygen uptake by the pulmonary capillaries and continual diffusion of carbon dioxide out of the pulmonary capillaries into the alveoli. So remember is that we have this soluble gas moving along a narrow pressure gradient from the blood into the lung, which is exerting its own pressure, right? So we've got carbon dioxide that's now exerting a greater component of the partial pressure of the total pressure within the lung. So this will result in a little bit less oxygen in the alveoli relative to the trachea. So now what happens is that we have an alveolar PO2 of about 100 millimeters of mercury, and the blood that's returning from the exercise tissues of a venous PO2 of about 40 millimeters of mercury. So now we have a pressure gradient for oxygen, diffusive oxygen flux of about 60 millimeters of mercury. And so under these circumstances, we will have diffusion of oxygen along its pressure gradient, along this pulmonary capillary, such that as it moves through the lung and becomes reoxygenated, when it leaves, in most cases, we have enough, the red blood cell spends enough time in the pulmonary capillaries to achieve full equilibration between the alveolar partial pressure of oxygen and arterial partial pressure of oxygen. So in other words, this desaturated blood coming in at a PO2 of about 40 millimeters of mercury, by the time it leaves the lung, has spent enough time in the pulmonary capillaries to result in a complete equilibration between oxygen between the lung and the blood. And so the arterial blood on average then has an arterial PO2 about equivalent to an alveolar PO2 of about 100 millimeters of mercury. So now we have arterial blood at about 100 millimeters of mercury moving towards the muscle. The muscle will then consume oxygen, and it will leave the muscle after having consumed a bunch of oxygen. Now it's returning back to the lung at about 40 millimeters of mercury. So again, we have the high energy component here of oxygen, diffusive oxygen flux from the blood into the muscle. And remember, at the onset of exercise, the intracellular PO2 will drop from about 40 millimeters of mercury, and it can decline all the way to 4 to 6 millimeters of mercury, which will facilitate that transport of oxygen from the blood to the cell to support oxidative phosphorylation. So now we have the transport of oxygen that will move back along the venous circulation. It will mix all together with all of the different PO2s from all the different draining systems, all the different tissues, and back to the lung for reoxygenation again. Carbon dioxide at the level of the tissue is producing carbon dioxide. So again, we're getting, say, a PCO2 of 46 millimeters of mercury. In this case, the carbon dioxide will transport through the venous system back to the lung, where the mixed venous PCO2 rather, will be higher than the alveolar PCO2, and you will have a concentration gradient in this case of just about 6 millimeters of mercury, because now you have an alveolar PCO2 of about 40 millimeters of mercury. But because CO2 is such a highly soluble gas, it doesn't require much in the way of a partial pressure gradient to facilitate rapid exchange of CO2 from the mixed venous blood into the lung, ultimately to be expired through expiration. Okay? So we have oxygen and carbon dioxide moving along their respective pressure gradients for oxygen delivery to the tissue and reoxygenation at the lung, and carbon dioxide production at the tissue and removal at the liver. So, how is oxygen and carbon dioxide transported? Well, the blood itself carries oxygen in two ways. One is in physical solution, and the second is combined to hemoglobin. So, in physical solution, this is really the fluid portion of the blood, which, as we know, is the plasma. And so, at an alveolar PO2 of 100 millimeters of mercury, which is normal and healthy, only 0.3 milliliters of oxygen dissolves in each 100 mLs of blood. So basically we have 3 milliliters of oxygen per liter of blood. This is only equivalent to approximately 2% of our total oxygen carrying capacity in the arterial blood. So the amount of oxygen dissolved in the plasma, which actually determines our arterial PO2 and has important influences on, through the oxyhemoglobin dissociation curve, the affinity of the red blood cell for oxygen, actually represents a very, very small component of the blood's carrying capacity for oxygen. So the majority of oxygen, therefore, is carried bound to the iron molecule of hemoglobin in the red blood cells. And so hemoglobin transports approximately 200 mLs of oxygen per liter of blood, equivalent to 98% of total oxygen carried per liter of arterial blood. So you can imagine then that you can appreciate that. I think this came up in your first lab, or it should have. Many of you, I'm sure, discussed it in your lab reports, where you're talking about the potential differences in oxygen carrying capacity between men and women. If men are generally bigger in body size, they have a bigger blood volume. So for a given, say, like for a given amount of hemoglobin, more blood means more oxygen carrying capacity. And so for a given cardiac output with higher oxygen carrying capacity, you should be able to support a higher absolute VO2. So again, something bigger that has more blood has technically a greater oxygen carrying capacity than something that's small. Again, so this is just kind of a volume related effect. Okay? So the oxygen carrying capacity then is equal to the concentration of hemoglobin multiplied by the oxygen carrying capacity of hemoglobin. So an average healthy adult has a hemoglobin concentration of about 15 grams per 100 mLs of blood, and each gram of hemoglobin binds with approximately 1.35 mLs of oxygen. So the oxygen carrying capacity then, if you calculate it out, 15 grams of hemoglobin per 100 mLs of blood times a carrying capacity, of 1.35 mLs of oxygen per gram of hemoglobin gives you about 20 mLs of oxygen per 100 mLs of blood, which is equivalent to about 200 mLs of oxygen per liter of blood in terms of our carrying capacity. So, again, you can appreciate that if you had a higher blood volume at a given hemoglobin concentration, you would have a higher oxygen carrying capacity. If you had a circumstance where your hemoglobin levels were low, you were chronically anemic, for example, which happens in a lot of conditions, you'd have a compromised oxygen carrying capacity. Women, throughout their menstrual cycle, have loss of blood and iron-containing molecules, and as a consequence, there are potential performance-related differences related to menstrual cycle phases because of compromisation in oxygen carrying capacity. Okay. So, oxyhemoglobin dissociation curve, and really describing the relationship between the partial pressure of oxygen dissolved in the blood and the amount of oxygen bound to hemoglobin, or the saturation curve. So we have the percent oxyhemoglobin saturation on the y-axis and the partial pressure of oxygen on the x. And much like the pressure volume curve of the respiratory system, we kind of had this sigmoid relationship of percent saturation and the partial pressure of oxygen. And so we know that at very high alveolar and arterial blood PO2s, we have a very high affinity of hemoglobin for oxygen, and therefore we have a very high percent saturation. Whereas we move down this oxyhemoglobin dissociation curve towards lower PO2s, we move along the steeper portion of the curve, more along the mixed venous PO2 level, right? So about 40 millimeters of mercury. And so under these circumstances, at the upper alinear portion of this curve, you can appreciate that we can have relatively large swings or changes in the arterial PO2 without compromising much in the way of percent oxygen saturation, or the amount of oxygen that's found in the hemoglobin. So again, we can have some disturbance in the arterial PO2 without really compromising much in the way of oxyhemoglobin saturation. And so you can appreciate, though, that the high partial pressure of oxygen in the lungs increases the affinity for hemoglobin for oxygen, which actually facilitates the binding of oxygen with hemoglobin for transport to the tissues. So the high PO2 at the level of the lungs basically will allow for that oxygen that's moving across the lung-blood barrier to really bind aggressively to the hemoglobin to subsequently be delivered to the exercising tissues. So it basically permits loading of the red blood cells at the level of the lung. And so we're occurring along this oxygen transport cascade at a PO2 here around the alveolar gas, in this case of about 80 to 100 millimeters of mercury. Alternatively, at these lower PO2s, which are more consistent with the mixed venous blood, is that we have low PO2s in the tissues, again, which are really a reflection of what the mixed venous blood is, ultimately decreases the affinity of hemoglobin for oxygen, and this will facilitate the unloading. So we don't kind of want to have these sticky hemoglobin molecules that are grabbing onto oxygen at the level of the tissue. We want to be able to have these hemoglobin molecules that are saying, you know, leave, go, transport to the cell to utilize in metabolism. And so ultimately, the low PO2s at the levels of the tissues and the mixed venous blood facilitates unloading of oxygen from hemoglobin to be used by the tissues in cellular respiration. So really, the body takes advantage of the shape of this sigmoid pressure volume curve. Oxyhemoglobin dissociation curve. So there are some factors that can change the shape of this oxyhemoglobin dissociation curve in the context of exercise. So during exercise, which is the example that we have here, so increases in body temperature, decreases in pH, or the development of systemic acidosis, so increases in hydrogen ion, or increases in this metabolite of glycolysis, so this 2,3-DPG is produced by red blood cells during glycolysis and binds with the heme subunits of hemoglobin, ultimately reducing the affinity for oxygen. The point is that exercise-induced changes in any one or combination of these factors can result in a rightward and slight downward shift of the oxyhemoglobin dissociation curve. And so the temperature and pH-induced shift in this curve is what is referred to as the Bohr effect. Okay, and so really, there's a facilitation or an unloading of oxygen during exercise because of temperature and pH-induced changes and shifts in the oxyhemoglobin dissociation curve. Such that, so you can see here is that at any given PO2, the rightward and downward shift of the oxyhemoglobin dissociation curve will serve to decrease the affinity of hemoglobin for oxygen and facilitate oxygen offloading. So the exercise-induced changes in body temperature and hydrogen ion levels actually facilitate greater unloading of oxygen at the level of the tissues, particularly at those PO2s that are occurring at those tissues. So it takes the steep portion of the curve and almost exaggerates it a little bit more, facilitating offloading of oxygen. The alternative is that if you have a circumstance where you have a decreased rate of glycolytic flux, reductions in body temperature, and a circumstance where you have an alkalosis, whether or not it's of respiratory origin or metabolic origin, you can appreciate that you would have a leftward and slight upward shift of the oxyhemoglobin dissociation curve, which would result in greater binding of oxygen to the hemoglobin molecules, which would try to protect oxygen exchange and oxygen saturation levels, such that again, at any given partial pressure of oxygen, you have a greater percent saturation. And so this will increase the affinity of hemoglobin for oxygen and decrease oxygen offloading. And so, for example, this is in part how hibernating bears are able to sustain their metabolism throughout the winter period, because they can really slow their metabolism and they take advantage of this upward and leftward shift of the oxyhemoglobin dissociation curve. So there's also transport of oxygen in the muscle through myoglobin. So myoglobin is an iron-containing protein that's found in skeletal and cardiac muscle fibers that shuttles oxygen from the muscle cell membrane into the mitochondria. And so high myoglobin concentrations are found in slow twitch or aerobic muscle fibers, and relatively low myoglobin concentrations are found in fast twitch or more anaerobic muscle fibers. Again, this makes intuitive sense. You'd want to have a circumstance where you'd have high myoglobin levels in high oxidative fibers and lower myoglobin levels in more glycolytic fibers. Myoglobin has 240 times greater affinity for oxygen than hemoglobin. So the myoglobin oxygen dissociation curve is much steeper than hemoglobin for PO2 values less than 20 millimeters of mercury. But this is actually advantageous because at the level of the mitochondria and at the level of the cell itself, we know that PO2 levels are actually very low, right? They drop from about 40 millimeters of mercury when they're in equilibration with about the mixed venous PO2, but during exercise we showed that they drop from 40 down to about 4 to 6 millimeters of mercury. And so at these very low PO2 levels, at the mixed venous PO2, you can see that myoglobin has a very, very high affinity for oxygen. But during exercise, when the PO2 levels at the level of the muscle cell drop rapidly down to 4 to 6 millimeters of mercury, you can see that myoglobin basically has no affinity for oxygen and just transports it and sends it to the muscle to be used by the cells. Okay, so it's a pretty effective oxygen transporter in that regard. So, carbon dioxide transport. Blood carries carbon dioxide from the tissues to the lungs in 3 ways: dissolved in plasma, bound to hemoglobin, and combined with water as bicarbonate that in a process that occurs within the red blood cell. So first, carbon dioxide dissolved in plasma. So we know that plasma will transport approximately 10% of CO2 produced in metabolism as unbound or free carbon dioxide in solution. So carbon dioxide produced by the cells will basically diffuse along the pressure gradient, dissolve into plasma, and be carried that way. The plasma CO2, much like the plasma PO2, is important because it establishes the partial pressure of carbon dioxide of the arterial blood. So again, if we're looking at in the lab, if we're measuring somebody's level of CO2 retention, this is actually what we're measuring. We're measuring in large part the partial pressure of oxygen dissolved, carbon dioxide dissolved in the plasma as the arterial PCO2. Second, a second important factor is the amount of carbon dioxide that's chemically bound to hemoglobin. So about 20% of carbon dioxide produced by metabolism binds directly to hemoglobin in the red blood cells. So you can see that carbon dioxide will bind to hemoglobin, producing this carbaminohemoglobin molecule. This carbaminohemoglobin molecule will dissociate in the lungs, so basically it'll transport from the veins to the lungs. And at the lungs, carbon dioxide will diffuse from the red blood cell to the plasma. Carbon dioxide will diffuse from plasma to the alveoli, and carbon dioxide will then ultimately be blown off. And again, this will occur because of the change in the partial pressure gradient that exists between the mixed venous PCO2 and the alveolar PO2. And so as hemoglobin comes in contact with the alveoli, the affinity for hemoglobin for carbon dioxide decreases. And this is in part because the affinity for hemoglobin for oxygen will increase at the alveolar PO2 levels according to that oxyhemoglobin dissociation curve. So basically what happens is that at the level of the tissue, there's not much in the way of competition between oxygen and carbon dioxide to bind to hemoglobin, because the hemoglobin are relatively desaturated, right? Oxygen is left. Whereas as it transports back towards the lungs and the alveolar PO2 rises, the affinity for hemoglobin for oxygen increases. The affinity for hemoglobin for carbon dioxide will decrease, and so they will basically exchange. Hemoglobin will take on oxygen and release carbon dioxide. Carbon dioxide will then be expired. Oxygen will load onto hemoglobin to subsequently be delivered back to the tissues. And this is what is known as the Haldane effect. So you will have to have an appreciation for differences between Haldane and Bohr. You can imagine a multiple choice question. The Haldane effect describes this. Is the right description to be true? Is the wrong description to be false? Right? So you have to have an appreciation for what the Haldane and the Bohr effects are. And finally, the majority of carbon dioxide is transported as bicarbonate. So ultimately, carbon dioxide that is produced at the levels of the tissue combines with water in the red blood cell in a reaction that's catalyzed by the enzyme carbonic anhydrase to produce this carbonic acid molecule. Carbonic acid is then transported through the blood. Once carbonic acid is produced, carbonic acid then dissociates into a bicarbonate ion and a hydrogen ion. The bicarbonate ion the hydrogen ion will be transported along the red blood cell, whereas the bicarbonate ion will diffuse out of the red blood cell into the plasma for transport throughout the body. Whereas, because you're losing a negatively charged ion from the cell to maintain the membrane kind of excitability and to maintain the cell as being electrochemically neutral, you have this chloride shift, whereas the bicarbonate ion leaves the cell, a chloride ion will shift into the cell to maintain this electrochemical balance. Right? So carbon dioxide produced at the cell will combine with water in the red blood cell, create carbonic acid, then dissociate into a hydrogen ion and a bicarb. Okay? At the levels of the lungs, that's the point. The point is that carbon dioxide is transported primarily in the blood as bicarbonate. So we will start the next class on Tuesday with the breathing pattern changes and moving into why do we breathe, the way that we breathe, what are the why is it optimal, and then ultimately moving into some experimental evidence of kind of muscle-lung competition for oxygen delivery. All right, have a good weekend.